This afternoon, I'm going to talk about some recent work that I've been doing with some of my group. As Ammar said, I got my PhD in 1990, and the topic of my thesis was modeling solid tumor growth, and that's been a main focus of my research since then. And some of the work in this talk, I'm going to take you back about 20 years to 2000 when I started looking at invasion work. And then this current work has developed from the invasion work that I did some years ago when I was still at Dundee University. So here's the overview. Before we get into the modeling and looking at metastatic spread, a little bit of background of what cancer is a disease and what metastasis is specifically within cancer, and how that what that is and how dangerous it is for patients. And then we'll see metastatic spread is very closely linked to invasion of cancer, so the local spread of cancer cells into the tissue. And so before we get to the metastatic spread, we'll have to look at invasion modeling, so cancer invasion of a local tissue. And then once that sort of groundwork has been made, we can then look at the recent modeling that we've done in the group in St Andrews on metastatic spread, which I'll explain in some detail. But metastatic spread basically means a primary tumor in one location then has the ability to move to multiple locations within the same host, which makes it difficult to treat. And then I'll touch on some future developments that we've taken on forward from the work on metastatic spread. So if we look at what cancer is a disease, it's a disease of the cell. Something goes wrong in the cell in terms of proliferative ability, and there's essentially excessive proliferative ability of cells, and that leads to the formation of, in terms of solid tumors, I'm going to focus completely on solid tumors. There are other tumors such as leukemias in the blood. They're completely different, as one might understand. So I'm going to focus on solid tumors. So due to excessive proliferation of these sort of mutated cells that are cancerous, you get lumps of cancerous tissues formed, which are solid tumors. And there are differences between tumor cells, cancer cells, and tumor tissue and normal tissue. And this was looked at in detail in a couple of famous papers now. The first paper appeared in the year 2000. And then an updated paper, as we can see here on the bottom, in 2011. And these papers are simply referred to for people who work in the area as Hanahan and Weinberg. And they're cited tens of thousands of times. And it's almost necessary if you're writing a paper on some aspect of cancer growth that you cite Hanahan and Weinberg. But anyway, Hanahan and Weinberg sort of classified the key differences between cancer and normality, cancer cells and normal cells, cancer tissue and normal tissue. And so in their initial paper in the year 2000, they focused on 6 things that were different between cancer and normal, and they called them the hallmarks. And in their 2011 paper, they brought in 4 other aspects, which I won't dwell upon. I'm going to focus on these sort of 6 initial hallmarks. And you can see here that there are 4 of these that occur at the sort of cellular level. That's resisting cell death. So cancer cells don't undergo apoptosis, which is the normal programmed cell death. So they don't die. They have a proliferative advantage, so they proliferate quickly. So they proliferate quickly and don't die. And they have sort of autocrine signaling mechanisms, which help them to resist death and proliferate. And they also have sort of escaped the body's normal control mechanisms. That's the evading the growth suppressors. So these happen either intracellularly or at the cell level, and focused on the cell cycle. There's a lot of work in that area. But at the sort of larger scales, 2 absolutely key aspects of cancer growth, which make it very difficult to treat, are the process of angiogenesis, which means the development of blood vessels towards the tumor. And I would have to spend a whole talk on angiogenesis itself. But basically, cancer cells secrete enzymes and blood vessels grow towards the tumor and oxygenate the tumor and allow the tumor to grow from quite a small size to a bigger size. And closely connected to angiogenesis is the process of invasion, which, as it says, the name implies, is the localized spread of cancer cells into the surroundings, which is the extracellular matrix. And then metastatic spread, which means spread to different parts of the body. Now, the metastatic spread via invasion is also connected to angiogenesis, because if you have an invading cancer, or if the blood vessels have connected to the cancer, then the cancer cells can get into the blood vessels and then be transported throughout the body, transported around the body, and then they also have the ability to exit the blood vessels at a different part and then get into new tissue at a secondary or a distant site, and then the whole process starts again. So rather than just one localized tumor, which is hard enough to deal with, you have then multiple tumors in different sites in the body, and that's very, very difficult to treat. There's also some interaction with the lymph system. I won't discuss that at all today, but that's similar to angiogenesis. Over recent last 5 years or so, experimentalists have got interested in the process of lymph angiogenesis, so the growth of lymph vessels to tumors. In some sense, it's quite similar. Blood vessels are, as the name implies, they're vessels that carry the flow of blood. Lymph vessels are similar vessels, although they're different structure, and they carry lymphatic fluid. The lymphatic fluid is different to blood, but in principle, you've got fluid in tubes, and that's interacting with the cancer cells. And both systems can take the cancer cells around the body. But as I say, today I'm going to focus on the two very closely linked processes of invasion and metastatic spread. And here's some information that everybody knows, but it's good to remind people, because I think most people know what cancer is, and probably we all know family and friends, unfortunately, who've had cancer and perhaps even not survived, which I'm sure we've all experienced. So people know that cancer is a big disease. It's, after heart disease, it's the second biggest killer in the developed world. And this is a definition from the World Health Organization, that cancer is a genetic term for a large group of diseases. There are malignant tumors, which really cause all the problems. Malignant tumors are called cancers. And one defining feature, so there's like the hallmark, is the creation of abnormal cells that grow beyond their usual boundaries. That's the definition of invasion. And then they spread to other parts of the body, and this is called metastasizing. And then there's the last sentence here that's really, really important, because a lot of people are unaware of this, that it's actually the metastatic spread, or the metastases, the secondary tumors, that are the real cause of death. If cancers remained localized and patients only had one tumor, that would be far more treatable, and there'd be far more success and survival if they remained as just one and localized, but unfortunately they don't. And this was also touched upon in the paper by Hanahan and Weinberg. This is a little section in there when they discuss specifically the hallmark of tissue invasion and metastasis. And here you can see that you have local invasion and you have new colonies. These distant settlements of tumor cells, metastases, are the cause of 90% of deaths due to cancer. So unfortunately, the metastatic spread is the real killer, because it's really very, very difficult to treat. And it's in multiple organs, and that's really bad news if you're a patient. Okay, so that's the sort of background to the biology of invasion and metastatic spread. So cancers don't remain localized, they invade their local tissue. And if you want to try and model this, we focus on the key variables in this invasive system. And there are basically three main variables. And you can make the models then more and more complicated if you want, but any invasion model should really have at least three variables, those being the cancer cells themselves, things called MDEs. So MDEs stands for matrix-degrading enzymes. These are enzymes that are either secreted by the cancer cells and they diffuse into the local tissue and then they degrade the tissue, create gaps. Or they can also be what's called membrane-bound matrix-degrading enzymes. And that means that they're secreted by the cancer cells and then they kind of rebound to receptors on the cancer cell surface. And once they're rebound, then they become active and the cancer, almost the cancer cell itself can degrade the matrix. So the cancer cells, either indirectly through secretion or directly through the membrane bound, interact with the components of the extracellular matrix, components such as collagen and fibronectin, and degrade the matrix. And then that allows the cancer cells through combinations of migration, active migration, and proliferation to spread into the surrounding tissue. So any model should contain these three components of cancer cells, MDE and ECM. And you can have different types of MDEs. There are lots of, a class of MDE is matrix metalloproteinases, and there are, up to 20 different types of metalloproteinases, all doing slightly different things in the matrix. So you could develop a model with several types of MDEs targeting several different components of ECM. But in what follows, we're going to try and keep things as simple as possible and just focus on a generic matrix-degrading enzyme and a generic tissue of the ECM. And if we look at the specific type of cell migration that's involved, this is known as haptotaxis. And haptotaxis, as the name suggests, if some of you are familiar in this area, it's like chemotaxis. So chemotaxis is a directed movement of cells in response to chemical gradients. So chemicals are secreted and form gradients, and then other agents can respond to those gradients, and essentially you move to a zone of high concentration. And so haptotaxis is similar to chemotaxis, except there's no diffusion of the chemical. It responds to gradients in the matrix itself. And the matrix doesn't diffuse, but it can be degraded, so you can get different densities in the matrix, and so the cancer cells will move to regions of higher matrix density in a directed manner. And so here's some images from an experiment that was done by some experimentalists looking into cancer invasion. And so these are some cancer cells, and this is on a glass slide. And because the cancer cells can't adhere to the glass slide, they remain localized and together. If you put some matrix components on the glass slide, then the cancer cells interact and adhere, and then use the differential adhesion present to start to spread out. So you can see now the cancer cells have moved away from the central mass. And then if you put a component that has really good adhesion, as you can see from this bottom image, the cancer cells are now spread completely across the whole glass plate, contrasting starkly with the first picture where they've all remained localized in the middle. So through this directed migration in response to variations in density of matrix components such as fibronectin and collagen, you get differential adhesion, and so you get this directed motion. So if you wanted to put together a basic model using these three variables with some PDEs, the reaction diffusion approach, so cell densities and matrix densities, here's a model which sort of captures all the key components. So we have our cancer cells. There's a little bit of diffusion. That random motility, that's small in comparison. This term here is our haptotaxis. So you can see the cancer cells are responding to the gradients in the variable V, which is our extracellular matrix. And there's some coefficient of haptotaxis rho. Excuse me. And then there's some cell proliferation, cancer cell proliferation in competition with the matrix, sort of a volume filling exercise. The matrix degrading enzyme is produced by the cancer cells, and that diffuses and has some natural decay. And then the matrix degrading enzyme, as it's diffusing through the tissue, the extracellular matrix, it degrades it at some rate, and if you want, there's perhaps some remodeling term which allows the matrix to get back to a healthy state if that's appropriate. So a simple, relatively simple reaction diffusion equation. You can, for those of you aware of these, this type of modeling, obviously with diffusion in a sort of logistic term, it's a variation on Fisher's equation with the extra sort of convection term of haptotaxis there. So one might expect some traveling wave solutions, and we'll see that just in a minute.
Mark, can I ask a question?
Yes, of course.
May I just know what the units of V is?
V is the density of the matrix, so it will be concentration per unit volume.
I see, and I guess C would be also concentration per unit area, I guess. As well.
So the cells will be also a density, exactly.
Density,
the number of cells per unit volume, or unit area if you're in 2D, or unit length if you're in 1D. Yeah, so these equations are all non-dimensionalized.
Okay.
I've got some references throughout the paper where you can look at the non-dimensionalization that we undertook. But as I said, just for the sake of the talk, any equations you see have all been non-dimensionalized or scaled beforehand.
I see, I see.
So if you want to make things even simpler, which is what we did 20 years ago when I was in Dundee. So this was joint work with Sandy Anderson, who's now in, probably some of you know Sandy, he's now in the Moffitt Cancer Research Institute, heading up the IMO, Institute for Mass Oncology. So Sandy and I worked with some clinicians at the local hospital in Dundee, Nine Wells Hospital, Luke Newman, Bob Steele, and Alistair Thompson. They were breast cancer surgeons. And they were interested in trying to work out how far a cancer spread into the tissue of their patients. Because surgery is still a viable treatment option if you get things early enough. But the question they were wanting to ask was, how far into the normal breast tissue does a breast cancer spread, and how would you detect the furthest distance that a cancer would penetrate into normal tissue? Because it's not always easy to work out from X-rays or CT scans just how far the cancer cells have penetrated. Because you can only take one or two of them, and if they get a little further away from, say, a visible margin, then the dilemma for the surgeons at that point in time, which was the question we were trying to answer, was what amount of tissue should you cut out to make sure that you were almost certain that all cancer cells were removed without doing taking out huge amounts of tissue. So it's a balance between being too conservative and just cutting around what's called the visible margin of the tumor, and then digging too deeply into healthy tissue and unnecessarily damaging the patient that way as well. And it only takes a few cells to remain after a surgical operation, and then some years down the line, the whole process would recur, unfortunately. And so what you want to do is to minimize that as best you can. So that was the reason for us working with the clinicians. And in order to do that, we felt that a PDE-based model wasn't really what we required. It wasn't accurate enough to detect these individual cells. And we'll see in a few slides' time just exactly how we did that. But we started off with a model. We discounted proliferation. We just wanted to focus on the migration of the cells, and in fact the influence of the haptotaxis. We from experimental data, you can show that the parameter rho is much bigger than the diffusion, and so this is like a advection convection dominated process in terms of the cancer cells. So these are some one-dimensional simulations we did, and as you can see from these early simulations, we had a one-dimensional domain, we had our extracellular matrix, and we had tumor cells, cancer in the left-hand part of the domain with some matrix-degrading enzymes. And then the secretion of enzymes, degradation of the tissue. You can see now there's a gradient being formed, and that allows haptotaxis to occur. And what you can see here happening is that you get a sort of a fraction of the tumor, almost, but not quite, it sort of breaks off, and you have some tumor left behind in the location it was. And this component of the tumor has broken off, and it's invading and penetrating deeply into the tissue. So sort of qualitative invasion metastatic break-off model, even with this simple one-dimensional simulation. And then you can put that into 2 dimensions. And so 20 years ago, this was kind of cutting edge stuff. So this is a 2-dimensional simulation, and we have some heterogeneity in the matrix. And so you can see that the white, which is the cancer cells, it's growing and invading, and it's sort of breaking up and becoming a heterogeneous mass. And obviously, from a point of view of surgery, this is a more complicated shape to try and cut out and resect than a simple sort of symmetrical, equally growing homogeneous mass. So again, even at the PDE level, the dynamics were showing some interesting things due to heterogeneity and sort of clumps of or regions of the cancer that are breaking off and forming little isolated islands. But of course, it's not, we don't just get breakup of little densities, little islands of cancer cells, where obviously with the PDE, this would really mean lots of cancer cells. It takes only handfuls of individual cancer cells to break off and remain sort of hidden in the tissue for the disease to recur if they're missed in surgery. So in order to try and capture the sort of individual cancer cell invasion, we developed a model that we heard of angiogenesis, which we developed to try and capture the shape of a blood vessel network, sort of discrete connected blood vessel network. And to do that, what we had developed in the late 90s, myself and Sandy, was that you look at the cell equation and you discretize that equation. Of course, you discretize it anyway if you're going to solve it numerically, and then you just plot the density. But rather than plot the density, once you've discretized the equation, what you do is you use that equation as a sort of probability movement generator for individual cells on a grid. And so for our cancer invasion model, this is our hybrid, we call it a hybrid continuum-discrete model. So we still have the equations governing the matrix-degrading enzymes and the extracellular matrix. But now our equation for the cancer cells is a discrete equation. And so cells now are on individual grid points, and this discrete equation is used to move cells from grid point to grid point. And if you're in a 2-dimensional domain and you have the usual sort of 5-point Laplacian stencil, 0 is your sort of central point, and then 1, 2, 3, 4 are up, down, left, and right, the grid points. And then these coefficients, P0, P1, P2, P3, and P4, which you generate from the discretized equations. And so in our equation, we have some diffusion. And you can see if we only had a diffusion equation, these 4 coefficients are equal, and that makes sense because for diffusion, it's an isotropic process, so movement in any one direction is equally likely. But now with the haptotaxis, you can see here, we have a sort of little gradient terms between the directions. And so these terms bias the coefficients P0, and there should be a P4 there as well. They bias them in terms of their size. And so we use this what we do at each time step of simulation is that these probabilities are now proportional to the probabilities of movement from one grid point to the next. And so we generate a random number between 0 and 1, having partitioned the interval between P0 to P1, and then P1 to P1 plus P2, etc. And so whatever interval your random number falls into, you pick that coefficient and then you move to that particular grid point. So if there are biases in the system, if you've got gradients in the system, you're more likely to move in a given direction. And so every time you generate a random number, that coefficient is the largest one. And so you're more likely to choose that. And so you're more likely to move in the given biased direction. And so that's how we generate cell migration. And then once we have this hybrid model of individual cells which can migrate from grid point to grid point, you can also add in some extra rules. For example, you can allow your cancer cells now to proliferate. So every 18, 20 hours, you can take a cancer cell and make 2 cancer cells and then put the 2 cancer cells on neighboring grid points. And then each cancer cell then interacts, secretes enzymes and migrates driven by the probabilities. You can allow cell mutation to occur. And it's known that as a cancer evolves over time, it becomes more aggressive, it becomes less adhesive to other cancer cells and more adhesive to the matrix. Proliferation might become more quicker, and other sort of individual phenotypic properties can change. And so you can add these things in at the individual level of each cell. So it's a sort of hybrid between a cellular automaton and a PDE. So all these other rules are added in, as I say, cell proliferation, differential cell-cell adhesion, differential cell matrix adhesion, and cell mutations. And so then if you simulate this discrete model, this is just showing you what happens to the cancer cells. You can see we start with a 2-dimensional domain. We have our extracellular matrix, matrix-degrading enzymes, and so the cancer then begins to spread and invade locally. And so you get what you would expect, things expand and invade the local tissue. And it was interesting to see that, for example, here, you can see that just because of the sort of inherent randomness and stochasticity in this sort of hybrid model, you can see here, for example, that one cancer cell is actually quite a distance away from the sort of main mass. And when Sandy and I took these simulations to our colleagues, our clinical colleagues at Ninewells Hospital, after we'd had an initial meeting with them and discussed the model, and we said we'll go away and work out the model and come back in a few weeks with some preliminary results. When we showed them these results, they thought we'd taken these results from some clinical paper of X-rays with metastatic spread. With local invasion spread, because they said this is exactly what they see in the tissue. And so they were very excited at this point. And at that point, we hoped to develop the model further into some sort of tool that they could use. But unfortunately, we didn't manage to get good enough data and develop the model in a sufficient way in order to help them explicitly. But they really could see the potential power of the model in making real predictions, or making some form of predictions based on real data that would help them in their clinical work. So we basically left the model at that point, as I said, because we didn't have enough data, and we moved on to other things. And then a few years ago, I had a PhD student who was interested in looking at some invasion models. And of course, in the interim, there's a lot more development in knowledge in terms of the underlying biology and what's been discovered in terms of the invasion as a process. And so my PhD student started off looking at to update the invasion model before we would maybe try and look at the metastatic spread. So in the intervening 20 years, 2 key things have come to light, and that one refers to what's known about invasion vis-a-vis the cancer cells, and there's now a very important process that people have looked at in detail called it's called EMT, and that stands for epithelial to mesenchymal transition, and that basically describes the invasion process at the cell level in some more detail. There are sort of 2 types of cells, and as you go and invade, cells change from one phenotype, epithelial, to another phenotype, mesenchymal, with slightly different properties, well, with different properties and performing different roles in the overall invasive process. So we thought that we should update the model to account for EMT. And then the other thing that had been discovered was that there's another class, a very important class of the matrix-degrading enzymes. They're not simply the diffusible proteins, metalloproteinases, but there's also a class of what are called membrane-bound proteins. And these are given the, so MMP stands for matrix-metalloproteinase or proteinase, and MT I can't remember the acronym there, but MT1 is the membrane brown thing, is membrane tethered or membrane tied. So these are molecules that are secreted by the cancer cell and then are rebound on receptors on the cancer cell surface. Here's the epithelial mesenchymal transition in a bit more detail. So a large number of solid tumors are called carcinomas, and they derive from epithelial tissue, which are sheets of epithelial cells. And so here's a little sheet of epithelial cells on a basement membrane, and the EMT occurs when you get this phenotypic change. And so your epithelial cells, which are largely inert and do some proliferation now and again, they change and become more motile and gain mesenchymal phenotypes and start to degrade the membrane, the basement membrane, and then migrate into whatever surroundings they have. If this is in experiments, then it would be in some kind of gel, and if it was in the body, this is the extracellular matrix. So the epithelial cells and the mesenchymal cells have different properties. Mesenchymal cells are more motile, they move, and it's the mesenchymal cells that are the active invaders, intravasate is the word used to describe how cells get into the blood vessels. So it's the mesenchymal cells that do the invasion, and they interact with the blood vessels and get inside. It's also the mesenchymal cells, because they're doing all this invasion, they're the ones that are connected with the matrix-degrading enzymes, both the diffusible ones and the membrane-bound ones. The epithelial cells, or the epithelial-like phenotypes, don't have any enzymes. They can't invade on their own. They require the presence of the mesenchymal cells. They sort of play follow my leader, and they're still very much connected in sort of sheets or columns of cells, and the mesenchymal cells are at the front and are broken off. They are a little bit motile, but not really when you compare them to the mesenchymal cells, but they are more proliferative. And there have been some models of this phenomenon in the last, say, 5 years. They're often called go or grow models. And so you take two PDEs and you have a PDE of mesenchymal-like cells, which are migratory, and then you have some transition between them and the epithelial cells, and the epithelial cells are more proliferative. And you can switch between the two, and again, there's some interesting traveling wave type solutions that are associated with such models. So these are called go and grow models, if you are interested, and look that up. Then on the matrix-degrading enzyme details, there's a very seminal paper published in 2009 by the group of Stephen Weiss at the University of Michigan. And his group in this paper really focused on these newly discovered membrane-bound matrix-degrading enzymes. And this is just a statement from the abstract from the paper. You don't need to read it all, except that this key phrase in bold, that they demonstrate in the paper that cancer cells have an absolute requirement for the membrane-anchored metalloproteinase, and that's MT1 MMP for invasion. So we'll just quickly look at the results of this paper. They had a very nice experiment. They took some cancer cells from spheroids that they'd grown, and then basically they put these spheroids into collagen gels, and they watched them grow over a period of time. And they did they had three options. They had a control experiment, which they just let grow, and because they were cancer cells, they grew and spread and invaded the gel over a period of 3 days. Then they took some cells and they knocked out in the cells the ability to secrete the diffusible matrix-degrading enzymes. So they only had the membrane-bound enzymes, and then they allowed the cells to grow over 3 days, and they looked at what happened. And then they did the opposite, and they knocked out all the membrane-bound MDEs and only kept the diffusible MDEs, and they watched what happened to that class of cells over 3 days. And so here are the results. On the left-hand column, we see their control. So we have the cancer cells in the middle, the red cells here. And then after 3 days, you can see they've invaded the local surroundings and into the gel quite extensively. In the middle column, this is what happens when you knock out the diffusible MDEs. and you can see there's almost no difference between the two outcomes. We have the same level of invasion. The distance is almost identical. And then in the last column, you see what happens when you knock out the membrane-bound matrix-degrading enzymes. Almost nothing. Now, they also stained for the diffusible enzymes, and they were present, but they have very little effect. There's a little bit of spread, but absolutely not nearly as much as when the membrane-bound is present. So they concluded that cancer invasion requires these membrane-bound metalloproteinase.
So how about the density of cells? Density of cells between these three experimental setup?
So I think that the density is the same, because I think they put the same number. You know, they counted out, I don't know, 2,000 cancer cells and put them in the middle. So the red bit here, these cancer cells, I think, are, you know, within experimental error, the same number of cells.
Yes, so but I'm talking about after invasion. I mean, the thing is, there you have these cells are migrating or spreading, but in the third condition, I'm wondering if cell division is not playing any. I'm guessing there's cell division as well, right?
There could well be. So I'd have to check the paper, but. Well, I mean, I think they discounted cell division as an option. This is all down to the localized migration.
Migration, okay.
I'd have to check the paper, but the cell division doesn't play much of a role at all.
Okay.
So yeah, the take-home message is that these membrane-bound metalloproteinases are critical for the invasive process. So we updated our PDE model. So we have we're going to discretize this again. So we'll put in all the proliferation at the discrete level. So we have now two cancer cell types: epithelial-like cells and mesenchymal-like cells. So CE and CM. So they're just a little bit of random motility and then haplotaxis. We have our diffusible enzymes, which we're going to call MMP2, matrix-metalloproteinase 2. So that's pretty much the same equation that we had previously, except here you can see that it's only produced by the mesenchymal cells, as the experimental data shows, but the MMP2 then diffuses and decays. And rather than explicitly include another enzyme that's secreted and then bound to receptors, because then there's complications with scales, what we assumed is that there's simply, when the cancer cells themselves, the mesenchymal cells, interact with the matrix, there's additional degradation. So, and in effect, that's what happens, because when the molecule's secreted and then bound to a receptor, it's as if the cancer cell, obviously it's bound to many receptors on the cancer cell surface, so it's as if the cancer cell itself is degrading the matrix. So this is the membrane-bound degradation, and this is the degradation due to the diffusible enzyme. And then we discretize the cancer cells equation, both epithelial and mesenchymal, in the same way. And we can put in proliferation, adhesion, mutation. And the other thing that we can put in is this transition from epithelial cells to mesenchymal cells. So there's a fraction of epithelial cells transform into mesenchymal cells, and then they can go and do the invasion and degradation of the matrix as per the previous equation. And the transition rate is determined from experimental data that people have looked into for various cell lines. So we, first of all, benchmarked our model. So this is the experiment of Weiss on the top, and this is our sort of benchmarking model. So we simulated our full system, and we got some invasive spread. We knocked out the diffusible enzyme and tuned the parameters so that we matched the data. And then when we knocked out the membrane-bound enzyme, as you can see, we still have a little bit of invasion, but it's far more localized in comparison when the diffusible ones are knocked out and the membrane-bound ones are present. So we just tune the parameters to match the data from the experiment of Weiss's group. Okay, so that was our sort of updated invasion model. And now, if we look at how invasion connects with metastatic spread, there's our primary tumor, here's the extracellular matrix, here's our local invasion, and here's our mesenchymal cell and some epithelial cells which are caught up with the mesenchymal cells in a little pocket of collective invasion, but the mesenchymal cells are undergoing single invasion. And then here's a nearby blood vessel. So this is what we now need to incorporate in the model to look at metastatic spread. So the local invasion occurs, and then when the cancer cells encounter a blood vessel, they can intravasate, and that has to happen through the mesenchymal cells. But when that happens, these sort of clusters of the epithelial cells which are joined to the mesenchymal cells, they can also intravasate, that's possible. And then when individual cells get into the blood vessel, then you can have, the CTC stands for circulating tumor cell. And so you can have these circulating tumor cells at a single level, or you can have them as clusters, both epithelial and mesenchymal, and they can do two things. They can either split into individual cells, mesenchymal and epithelial cells, or they can remain as clusters. So you have these options as when you're in a blood vessel. And then at some point, at some distant site in the body, you get the opposite process of intravasation, which is extravasation. So the cells reattach onto the endothelial cells of the blood vessels and then squeeze back through and into the new tissue, and then the whole process starts again. You seed your secondary tumor, your metastases, and then that starts to grow in the normal way with the invasion process that we had in the primary tumors. So this is our framework for the metastatic spread. So we take our basic invasion model, and then essentially what we do is we create a different multiple grids. So it's a multi-grid system. So we have a primary tumor on what we call a primary grid, and then we have the possibility of secondary tumors getting to secondary grids, and we have transfer between them. Our framework for this was that because we'll see this in a minute with the data. We envisaged that the primary site would be the breast, and then the secondary sites would be the lungs, the liver, and the bone, because it's known that breast cancer spreads to these three sites. It spreads to other sites, as we'll just see in a minute, but just to keep the simulations as efficient as possible, but still to get an idea. So we have a primary tumor and three potential secondary tumors located in three different types of tissue. And some years ago, there was some data gathered by some clinicians in the United States, in Houston, and they looked at almost 4,200 women with breast cancer, and they worked out the overall numbers of secondary tumors. So here you can see these are all the, you can go to the bone, lymph nodes, the chest wall, the lung, the liver, some other places, the brain, the kidney, the ovaries, and the skin. And so we incorporated this data as a probability to get to either the lung or the bone or the liver in our metastatic spread model, moving from the primary grid to the secondary grids. So we also then have to consider the interactions with the blood vessels. So on our invasion, on our primary tumor in the grid, what we needed to do was put some blood vessels and then account for intravasation. And then we had some blood vessels on the secondary grid, and that would account for the extravasation. We need to look at, there are two types of blood vessels. There are regular blood vessels and there are disrupted blood vessels, and cells interact with both in a slightly different way. Then we had to look in some detail as to what happens when cells, or these circulating tumor cells, are in the blood system, what happens to them there, and the probabilities of survival, the probabilities of disaggregation. There's a lot of very nice models of rheology and looking at these things and getting some nice data. And then, so once you put all these components into the initial invasion model, that would allow you to predict some metastatic spread. So here's our primary grid. And here's our primary tumor in the middle. This is our breast tissue with breast cancer, primary tumor there growing. And the red dots are blood vessels that have just been put on the grid and placed randomly. And we have, in addition to standard blood vessels, which are just red dots, we have two ruptured vessels. Something's gone wrong with them, and they're slightly different. And as I say, the cancer cells interact with ruptured vessels in a different way to standard vessels. And so what we're allowing this primary tumor to do is invade with our invasion model, and then when they interact with the vessels, we'll look at what happens using cellular automata rules. So this is the general sort of flowchart diagram for our metastatic spread. We have our primary grid with the primary tumor that invades according to the invasion model. And so we have proliferation and movement, and then the cancer cells spread to different grid points. If you do not encounter a blood vessel, you just continue moving up, you just continue invading. But if you encounter a blood vessel, so if you're a cancer cell and it invades at a time step and lands on a grid point with a blood vessel, you then interact with the blood vessel. And first of all, we say, will the cancer cell enter? There's a probability of entering, that's fine. If it enters into the blood vessel, there's a probability of survival. Again, we've got the data from the literature, so we allocate a survival probability. If it survives, then it can be transported. If it doesn't survive, it just dies. So if it survives in the vasculature, we let it survive for a certain period of time that you can estimate from data in the literature. And then we allow it to go to one of the three secondary grids. And then we give it a probability of extravasation. And then once it extravasates into the secondary grid, we allow it to spread. So, just then, with my eye on the timer, I've basically described this. So we have cellular automata rules for intravasation, getting into the blood vessels, both normal and ruptured, and that's focused on the mesenchymal cells. There are also clusters, so they can get in as well. So they were saying, yes, single mesenchymal cells can get in, but epithelial mesenchymal cells, which are in a cluster, can also get in. And then once they're in, they disseminate in the blood vessel if they survive, and then we allow them to travel through the vasculature, having survived for a certain number of time steps, and then you can extravasate at the secondary grids, and then once you're into the healthy tissue, then that's it, you can start invading according to our hybrid model at the secondary grid. So that's it. When the cancer cells reach the secondary grid and are successfully extravasated, they can then begin to spread and grow according to the original invasion model. So this flow chart here, as I say, in summary, describes this little schematic of the primary tumor, local invasion, interaction with a blood vessel, intravasation, survival in the blood vessel system, and then extravasation at a secondary site, and then setting up a secondary tumor in the secondary site. So here's our sort of simulation results. At the primary site in the breast, we have mesenchymal cells and epithelial cells, and they grow and invade according to the discrete model, hybrid model. And as you can see, the epithelial cells tend to remain in a sort of mass, whereas the mesenchymal cells are the ones that get to the blood vessels first. There's more or less on the, well, there are some in the middle, but if you superimpose these, then the mesenchymal cells are on the outside and the epithelial cells are sort of following them. Here's the diffusible MDEs, and here's the degradation of the matrix, which is sort of mirrors the cancer cells. And then if we go to the secondary grids, these are some snapshots at an early time and a later time. As you can see here in the lungs, we have some epithelial cells appearing at a relatively early stage with no mesenchymal cells, and that corresponds to the data. And then these epithelial cells begin to grow and take place, and we get some mesenchymal cells then following up after, which again is observed from the data. In the bones, we have a similar situation, except there's less spread to the bone. So at an early stage, there's not much is observed. There's nothing is observed at all, in fact. And then at a later stage, at a later time, we have some epithelial cells managing to get into the bones, and we have a smaller number of mesenchymal cells then following also. And then for the liver, although there doesn't appear to be much happening in either here, at an early stage, nothing has happened. But if you look down here at this little blood vessel at a later time in the liver, we see the beginnings of some mesenchymal cells have managed to extravasate and get into the tissue and start to spread. And again, that's what the data shows, that it's mesenchymal cells that get in the liver first. And then if we run the simulation for longer, the mesenchymal cells take hold and we do get some epithelial cells coming after. So just a little summary of the simulation results that were connected back to either the observed data or some in vitro work, that the shape and metastatic distribution were connected with what the data showed in untreated cancer patients. Obviously, what happens is if people start to find, if a patient is discovered at an early stage, they're treated. They don't, they're not treated as a sort of mouse and brought back and you look at it. But some patients are only found to have this at a late stage, and so you get late stage data. As I say, the mesenchymal cancer cells were they were all around the front at a leading invasive edge, and this has been observed experimentally in vitro work. When we looked at the invasion speed, that correlated nicely with these experiments of this group back in 2009, and we did see the correct types of cells seeding the secondary sites of the lung, the bone, and the liver in the correct manner. Of course, that correlates with the data from the US group with the 4,200 breast patients as to where what seeds where. And so that's all reflected in the computational simulations. So qualitatively interesting results and qualitatively correct results. And what's now needed is we need to work with some more data, try and refine the model a little bit with some of the things that happen with the cells. And it's our hope that in developing this, we'd like to create a so-called hybrid framework where rather like our aspirations 20 years ago for the local invasion, trying to help clinicians with surgical removal, if we have individual patient data, we could perhaps run this model and make some predictions to clinicians on quite a short time scale as to the likelihood of spread, and that might affect the way treatment is. If you were likely to spread it on a longer time scale, then you might want one type of treatment. If the model was predicting that the spread was going to occur over a short time period or go to multiple sites over a short time period, then clearly you would want to adopt a different treatment protocol and adopt that on a far faster time scale. Our other hopes for the modeling is to link this with more force-based models. At the moment, it's just a reaction diffusion system and probabilities of movement. And so when you get down to the cell level, interactions with the matrix, of course, in the phenotypic changes between epithelial and mesenchymal, there's a lot of interesting biomechanics involved. So we have some biomechanical models of cell migration and blood vessels. And so we're going to try and fuse together the two approaches into sort of biomechanical reaction diffusion system. And also try and link it within one of our angiogenesis models where we get explicit flow in the system with the cancer cells, rather than just a probability of survival and then or a probability of death. And then just in the last few minutes, I'll just quickly some further slides. Well, first of all, to say that this work has appeared in the Bulletin of Mathematical Biology online last year, just the end of last year. And I'll take this opportunity to thank my PhD student, Linnea Franson, who did a lot of the hard work, and two co-workers, Tommaso Lorenzi and Andrew Burgess in St. Andrews. And then a very recent paper with Linnea, just accepted a couple of months ago in the IMA Journal of Applied Maths. So Linnea looked at a sort of reverse transition. So as well as EMT, you get MET, so mesenchymal epithelial transition. The mesenchymal cells can sometimes go back to epithelial cells and become less invasive. So we've published some recent work, say in the IMA Journal. That's accepted a couple of months ago. That should be appearing quite soon. And then to try and connect with data, what we've done is we've, in a different direction, we've developed a new hybrid continuum-discrete model, and we've started applying that to organotypic assay modeling, which there's a lot of very good data there. This is, I'll describe this in just 2 seconds. So the new hybrid model has just been published in the SIAM journal, Multiscale Modeling and Simulation. It's a little bit technical, and I don't have time at all other than to say, if you're interested, please have a look. We have a PDE approach to the invasion, and then we have an interesting transition probability to take us to a stochastic discrete system, which allows us to move between groups of epithelial cells or densities of epithelial mesenchymal clusters and individual mesenchymal cells in an interesting, more sophisticated way than simply discretizing. And we've very recently applied that to look at some organotypic assays. These are assays which you have collagen gel and some cancer cells on the top, and then you can very nicely look at the cells as they invade and make very accurate measurements of how far and how aggressive the cancers are at invading. And so you can get a lot of nice parameters for an invasive model such as ours. And so yeah, they do a lot of model measurements. Of course, this is a 3-dimensional gel, but they then take a 2-dimensional slice. And so these are cancer cells on the top, and then they start to invade. And you can see in some cases the invasion happens with little clusters, and then in other cases you get far bigger clusters of cells that have broken off the main body of cells on the top of the matrix. And with our, this is very recent work, we're just about to submit it. This is our hybrid continuum discrete model. So we're trying to match as closely as we can the invasive assays, these organotypic assays. And this is work with colleague Nikos Sfakianakis. And Nikos is a numerical analyst, and he's managed to do this in 3 dimensions. So this is my last slide. So it's very easy for us to take a 2-dimensional slice and compare with the 2-dimensional data. But as you can see here, the yellow is the cell density, that's from the PDE model, and you can see the red, those are individual mesenchymal cells. And we've transitioned between the PDE model into these discrete cells with a technique we describe in the paper. But with this approach, we've been able to get some really nice matching to their data, and we've done lots of sensitivity analysis. And with this 3-dimensional model, it's very easy for us to really get an insight into the full 3-dimensional nature of the invasion, as well as taking 2-dimensional slices and comparing with the data that's been published in some papers. So I'll leave that there, and I'm very happy to take any questions from you.
Thanks very much, Mark, for a great talk. So the floor is now open for questions. If anyone has questions, I'll keep mine until the end, but please to ask questions. Actually, there's one that is written in the chat room by Nikos. Do you want to say it yourself, Nikos, or shall I read it?
Hello, can you hear me?
Yes, yeah, go ahead.
Thank you. Very nice talk. Thank you, Professor Chaplain. I would like to ask you for slide 23, why there is no growth term in the PD for cancer cells?
Yeah, so so this is simply so the original invasion model that I had in 2000 with Sandy Anderson and the clinical colleagues, what we did was we chose to try and focus on the effect of haptotaxis. In the paper, we changed the haptotaxis coefficient, and we actually saw some interesting dynamics on a purely migratory level. It's a little bit sort of theoretical, but it showed us the importance of the haptotaxis parameter. Clearly, there is cell proliferation, but so what we do, sorry I didn't explain this or it didn't come across clear, is that when we discretize these equations, we get an equation for discrete cells on the grid, so the cells can migrate from grid point to grid point, but at the discrete level, we also include cell proliferation. So every 20 hours or whatever, however fast the cells are proliferating, we can allow the cancer cells to divide into two. And so then each cancer cell then starts to invade and secrete enzymes. So we do include proliferation, but it's at the discrete level.
Okay, can I, since you are on that slide, just want to ask you a question about the P4. I mean, the P not all the way to P3 depended on diffusion and also the gradient in V. But for P4, you're actually in the same position. You're not moving. So how do you define P4?
I've probably chopped that off.
Yeah, in that slide. So that's why I'm a little bit puzzled how you define it.
P4 will not have any. So P4, so P4 doesn't have any gradient terms. Yeah.
Okay.
Okay.
That's a mistake there. Sorry. Yeah.
Mike, that's interesting. I was just wondering, density-dependent diffusion.
Yeah.
Must play a crucial role. And when I was thinking about some of the basic models, I know your models later have convective terms, and probably they're looking at, for example, the equation you've got up there. I guess the CE you sort of work out across the grid, right? So it sort of varies. It's not a constant. But I was thinking with the basic sort of diffusion model, we know from parabolic equations that you have an infinite speed of propagation.
Yeah.
And so I guess this gets around it, right? So you get finite propagation speeds, and by putting in convection and.
Well, that's a good point, but in fact, Nikos Sfakianakis, he had a summer student. He had a project student this year with him. And he actually got the project student to look at nonlinear diffusion in these invasive models for exactly that reason. Yes, so right. He has some nice results, and obviously at a PDE level that makes a lot more sense, because if you put if you have the diffusion proportional to the cancer cells themselves, so like C squared or something,
right.
C squared grad C, then you have compact support, etc., etc. So. All that's there. I mean, because we're using these PDEs essentially to generate the discrete cells, the issue of infinite speed and densities everywhere doesn't really arise.
Right. Yeah. So interestingly, I mean, what is mechanical forces? Everybody now. I mean, you're sort of trying to incorporate that in a sense. You have some of that really present with the haptotaxis type terms, right? But
yes.
But I like the idea of the multigrid thing, because you do have mechanotransduction and information that's transferred down and reversed. So with this hybrid continuum model, I did look at your earlier stuff, but now it sort of slips my mind. But I was just thinking. If you're looking at a centimeter cube tumor, you're looking at about 10 to the 9 billion cells or so. So when you do the discrete thing, are you looking at something like a fluid particle? So your particles are not really cells themselves, but a cluster of cells rather than.
So no, that's a very good point. I mean, yes, it depends on the scales. I mean, I perhaps should have also said this. The tissue scales we're working at is about 2 millimeters. So it's because I mean, if you need a supercomputer to simulate a cubic centimeter of tissue, which well, I actually, for a period of time, had collaborations with some colleagues in the University of Warsaw, and they did have access to a supercomputer, and they actually did manage to simulate up to a billion cells with a four-space model.
Okay. Okay. So you do want to stick with cells rather than, I was sort of thinking, can you think of it like a fluid particle where you have, you know, so you may have a cluster of cells which would behave.
We've adopted that approach in other models. So let's say, some people like to call these agent-based models rather than individual, so that's fine. So you can have an agent represent a number of cells rather than one single cell. And then you can, then it's obviously computationally feasible, and you can argue that you can simulate, you know, tens or hundreds of thousands of individual cells with this sort of meta model.
Right. I've got a couple more questions, but maybe if anybody else has got some, I'll come back rather than monopolize.
Who has other questions before?
I have questions.
Go ahead.
Hi, Leon.
Hey, Leon.
Hi, Mark. It's been a long time. It's a weird.
Yeah, lovely to see you. Really nice to see you.
Yeah, I'm still around. Thanks a lot. I was interested in metastasis a really long time ago, and there were a couple of questions that I found really interesting, which I'm not, this is more kind of to get an update after 40 years or whatever it is, but kind of getting to some of it right at the end. So one of the questions that I found fascinating was what's actually the spatial distribution of metastases in organs. So for some organs, it may be rather hard to measure that, but other ones maybe not, and also with different imaging technologies now with various active materials. So if there's metastasis in the liver or the lung, for example, how many metastases are you? What are they spatially located with respect to one another? So that was one kind of question. Another question is related to the sites of metastasis, and some tumors have preferentially go to one organ rather than another. I know that back a long time ago, that seemed to be a big mystery. People didn't really understand it. And I don't know if they understand it better now than they did then. And the third thing is the time scale. So I know that some of the things seem to have realistic times, and other times you said, well, everything is dimensionalized. So who knows what the space is and what the time is. But in things like breast cancer, the time scale for metastases can literally be years, if not tens of years. So could you just comment on those three things? One is the spatial distribution of metastases, another is the preferred organ, and the last is the time scale.
So first of all, before what we found is that when we started this, is that in terms of modeling, there are very, very, very few models of metastatic spread. There are some one or two models of statistics, you know, trying to work out probabilities. But so this is one of the reasons that we wanted to do this, because it's in terms of, as I say, modeling, it's very much under looked at. It's not looked at all. So we hope this will generate some interest in the community, and people will start to pick up on that. In terms of the spatial distribution in organs, yeah, I mean, I'm aware for example, the brain, if you get a brain tumor, that's it. You just got one horrible brain tumor as a secondary, and that's it. You don't get multiple brain tumors. But in the liver, you can get multiple nodes in your liver. We didn't really look at that. So that's something that we probably could look at again, or somebody else could look at. But yes, in some organs, you get sort of multiple foci of these secondary tumors. In terms of the preferences, again, we didn't get into that in detail because we chose to focus on a primary breast with the data from the Houston group with the 4,200 breasts, where they'd simply said all the possible secondary locations, and we chose the three favorite secondary locations for breast cancer spread: the liver, the lung, and the bones. but yes, there are primary tumors which have preferential spread into other organs. We'd have to go back and look into the literature and find that out. In terms of the timings, so yeah, we worked on it. Our scalings were a 2 millimeter domain of tissue, and I'd have to look up our non-dimensional timescale, but our timescale did correlate to experimental data of cancer cell growth, sort of in vitro invasion models. Now, the reason for long time scale, dormant metastases and things like that, there are a whole range of issues that come into play. One of which is the immune system. It's thought that the immune system can suppress things for a long period of time, and then something suddenly clicks and you get a metastatic spread. Some years ago, there was some thinking that anti-angiogenesis factors were involved. There's some very nice experiments from about 15 years ago where if you take a mice with a primary, I think it was a lung tumor, and you have a strong angiogenic response, and so your primary tumor in the lung is growing, and you do nothing, obviously the mouse dies. And then if you say, okay, let's take out the lung tumor in the mouse, what they found in a large number of these mice is you suddenly started to see these metastases springing up. They were called occult metastases. And the thinking was that anti-angiogenic factors were at play in the secondary tumors, and that suppressed the secondary growth. And so I was aware of some work in the literature at that time, but again, we didn't look in detail at these other aspects of dormancy and suppression and longer timescale metastatic spread. But that's definitely there, and would probably involve other realities such as I say, certainly the immune response will have a role to play. I hope that answers the question to some degree.
I think that the answers are that the questions that fascinated me then still are not very well studied.
Absolutely not. No, that's right. There's still a lot to be discovered.
The spatial distribution of tumor in the liver, I mean, we went so far back 40 years ago to visit the morgue to talk to people about trying to do serial slices through livers to try to see where the tumors were and what's their size, how were they distributed with respect to one another, how many there were, and so forth. And of course, nobody would fund us money to do anything like that. And the guy was collaborating with left. But these things now with imaging technologies are completely doable as far as I think. So they have, you know, sugars or whatever that are radioactive and show up in rapidly growing tissues. So I think they could get a very good idea of where tumors are, how big they are, and they could do that serially. And that's what metastasis is. So it's just interesting that that a lot of those questions seem to be open. And I think it's terrific that you're pointing the direction towards looking at metastasis, because that is definitely what kills people. So thanks a lot.
Oh, no, thank you. Thank you.
Yeah, Mark, just to follow up on, you know, you talked about the epithelial mesenchymal transition, and that obviously gives you the mechanism by which the primary tumor goes. And then when it extravasates, it goes through the mesenchymal epithelial transition. Now, I remember seeing Weinberg, some of Weinberg's work sort of saying that the mesenchymal phenotype ties in very closely with the cancer stem cell phenotype. And, you know, I guess Hanahan and, you know, they're sort of the cancer stem cell hypothesis, saying that this is what drives tumor growth. You know, so modeling efforts toward that direction of sort of trying to model it in terms of the cancer stem cell hypothesis. I know I talked to John Dick a while back, and he was saying, well, they don't believe it's just primarily that drives, because plasticity also plays a role. So you've got a number of things. So just wondering whether you thought about incorporating that type of thing, the cancer stem cell, as a sort of small population primarily driving tumor growth. And
yeah, I mean, so I published, well, I I was co-author on a paper some years ago with Heiko Enderling.
Mm-hmm.
And Heiko was very interested in cancer stem cells at that point. And he did an agent-based model and made certain assumptions, and it was about radiotherapy treatment. And so we grew some, we did some simulations with some assumptions on proportions of cancer stem cells and what they did, and what did the daughter cells die. I can't remember the details. But again, we got some nice results when you treated the tumors with different proportions of cancer stem cells in them. We got quite different results. And so I think certainly one for me, one nice thing about this framework is that since you're working with discrete cancer cells, you've got a lot of flexibility. So to incorporate some rules for cancer stem cells wouldn't be too difficult. As some other as maybe a third population, or the population that seeds the epithelial cell or something like that. So yeah, I think this framework offers a lot of flexibility for people to pick up on and look into ways that they're interested in.
Right. So by the same token, I like the multi-scale thing, because there's obviously information transfer across all scales, and this sort of captures it. But we looked some time back at the sort of poroelastic type modeling of tumors, and now when you were talking about blood vessel distribution, I was wondering, if you think there's any role for, say, a triphasic type model, where you take, you take the sort of poroelastic thing, but now add the blood vessels and just homogenize over the three phases. I know that's not going to be multi-scale, but maybe that could sort of give some indications of blood vessel distributions, etc.
Yeah. I mean, I think over the years, I mean, just my background in preferring PDEs and all that, but I think. I mean, I've got, you probably know him, Luigi Preziosi. I mean,
I was thinking,
Luigi does a lot of lovely work from a biomechanical point of view. And we've talked over the years, and it would be really nice to combine the best aspects of both. But I think that, again, if you're looking at biomechanical approaches, I think, I mean, I've not worked a lot on biphasic stuff. I think Helen Byrne and Luigi have done some really nice work in there. And of course, probably you do one's done the easy thing yourselves. If nobody's worked in the field, you take what's known from another field and and then call it a biphasic cancer model. So, and that's good up to a point. And then once other things come along and more information is known, you think, okay, well, there's a limitation. We need to make it. So a triphasic thing with blood vessel information would seem a nice way to go. So
you're right. Philip was saying as well last time when he was talking that you have all these reaction diffusion models, but it's becoming quite clear that you have to have some biomechanics in there, and the so those are So anyway, it's interesting times at least.
Oh, absolutely, yeah.
Anyway, thanks a lot.
Any other question? Okay, let me ask mine. Just one more question. The later model, the second version of this model that you're showing here on the slide, you talk about epithelial cells turning into mesenchymal cells. But then the last, the extra slides that you had towards the end, you talked about also back transition, going from mesenchymal to epithelial. And this is the question I had in my mind the whole time. Wouldn't that actually mechanism alone be sufficient to generate these colonies that you're calling it clusters? You're ending up with this, you have mesenchymal cells that are migrating, then they turn into epithelial cells, and that generates a new cluster. Wouldn't that mechanism be sufficient to do that kind of, produce that kind of behavior?
Yes, it would, but that's not really what the data shows. I mean, the data shows genuine so there is it's a genuine two-way thing. Although the MET is not as it doesn't happen as frequently as the EMT. So I mean, it's not all driven by things just flipping. And mesenchymal cells getting to the place and then reverting back to epithelial cells. And so there are genuine epithelial cells, because the people have done some experiments where they've tagged things, and it's clear that you get epithelial cells from the primary in the secondary, and it's not.
The reason I'm asking that question is because we are interested in adhesion dynamics. We're talking just about now about the mechanics of the cell processes, and we're modeling adhesion dynamics and how they regulate cell migration. When I think about epithelial cells, I think of them as being possessing stable adhesions. They block cell motility. That's why they're actually stuck where they are. So that's the reason I'm thinking that if you have that transition back, and of course you have cell-to-cell adhesion as well, so that actually might drive the formation of these clusters. Wouldn't that be proper kind of thinking about this system?
Well, yeah, actually, maybe I can just jump in. I think in Mark's case, the epithelial mesenchymal transition gives you the mechanism by which the primary tumor gets to a secondary location. Now, once it extravasates there, then you want the reversion back to epithelial phenotype for it to be able to drive a tumor growth, because the phenotype, I mean, the mesenchymal is just much more motile in a sense. Yeah. So I think that's am I right there, Mark? Or?
No, you're right. I mean, if you get lots and lots of mesenchymal cells, they don't proliferate very much. So if they've just come out of if you imagine them just coming out of a blood vessel, they'll just, in order to increase their numbers, you'll just have to have more and more of them coming out of the blood vessel. They don't really proliferate that much. So either you have to get epithelial cells in through the intravasation, through the vessel and extravasation, or then you take an MET, and then some of your mesenchymal cells at the secondary sites become epithelial-like, and then they can proliferate, and then you get a mass. Now, so I'd have to look at the recent paper and look at what we were doing, but it's probably a mixture of the two. What a surprise. But yeah, you could probably push it to the limit and say, okay, let's just look at extreme cases where the mesenchymal cells then, no epithelial cells get to the secondary locations, and any epithelial cells have to come from an MET transition, and see if you can connect that with the data. I mean, I'm trying to recall now, I think Linnea, my PhD student, she did some of these things. She looked at proportions of cells in each population and tried to show the best match to the data.
Yeah. So my impression is that in the cluster, it's really the mesenchymal. I mean, you have a cluster that sometimes goes in, but the proportion of epithelial cells in there is very small, right? I mean, you don't have that mesenchymal phenotype, you're just not going to be able to drive, get into the blood vessels. And likewise, as you said, at the other end, when they get out, unless you revert to.
Yeah.
Okay, thank you. Any other questions? Okay, so thank you very much, Mark, for giving us this insightful talk. That was really exciting work, and hope to see you again soon.
Yes, well, I hope it's face-to-face next time.